Okay, so as Florent just explained, plant population response to global change can imply either local extinction of population. They can imply migration, meaning eventually range shift for at the species scale, and they can locally adapt to new environments. Now, quaternary ecologists have studied these responses, these plant responses to past climate change, especially the glacial, interglacial cycles. And from the 20th century paleopollen records, a major, conclusion was that range shift was the only consequential, the only consequential response to quaternary climate change. I discuss elsewhere the key role of natural selection and local adaptation to past climate change. But this is not the topic of this talk, so I encourage you to read these, of course. But for now and for the remainder of the presentation, let's go back to migration and range shift. So these pollen records about the post-glacial spruce colonization, what they informed us is that it probably, like inference on black spruce colonization, probably show a dispersal speed or migration speed above 400 meters per year. However, this speed is probably too fast because spruce estimated maximal dispersal speed was thought to be below 100 meters per year. So this discrepancy between inferences from pollen records and modeled dispersal speed was called the Reed's paradox of rapid plant migration. So nowadays, in the 21st century, we have, yes, we still have the pollen records, but we have microfossils, wood charcoal, species distribution modeling, phylogeography, and we are using rather an integrative approach with these different proxies to create joint inferences about past plant responses. And using such an approach, we were able to detect what we've called cryptic refugia. These refugia were located closer to the ice sheet than previously thought or than previously inferred from pollen records alone. And for spruce, with respect to spruce colonization speed, we're now able to revise it down to 40 meters per year. However, this, of course, explains Reed's paradox of rapid plant migration, but it's rather bad news because forecasted boreal climate change velocity should be around 400 meters per year. So the existence of cryptic glacial refugia forced us to revise downwards plant migration capacity. Now, what does this mean for future climate change, ongoing climate change, anthropogenic climate change, if you will? So if these data stands, well, we might expect broad-scale migration lags if species cannot track the velocity of current climate change. And this is especially true in a fragmented landscape that was never seen before. It's an unprecedented fragmented landscape due to, of course, land use changes, anthropogenic land use changes. So these two fears, if you will, are or were discussed in multiple papers, and I just show a couple of these here. I will not review these papers at this moment. Rather, I will focus on a few silver linings, right? That's a tale of three conifers, my tale of three conifers. So first of all, the tale of tamarack. Tamarack is also called eastern larch, Larix laricina, and you see here the range of eastern larch. It's a transcontinental species across boreal North America, and you see, however, that there's a huge gap in the range across the mountains in Yukon. So what we've asked is, did larch persist in Alaska throughout the last glacial maximum? Or did it migrate from south of the ice sheet, from a well-known refugium, major refugium south of the ice sheet? So persistence, local persistence, or migration from the south. So first I start by showing you a species distribution model of the present to validate that our species distribution model is sound or is robust. We compare it to the current range and it's fairly coherent with the present range, you see. And what is important or interesting is that we've hindcasted past climate suitability using this model projected on last glacial maximum climate. And what we see is in fact that last glacial maximum climate was indeed suitable for large persistent. This is no proof that the large was present in Alaska, but it shows that the climate was suitable. Now, to assess whether larch was indeed present in Alaska, we did DNA analysis. We relied on 3 chloroplastic DNA markers from 69 populations across the range. And we found actually 3 distinct genetic lineage when we cluster all this genetic variability in lineages, one of which is endemic to Alaska. And this indicates, the yellow one, and this indicates glacial persistence in a cryptic northern refugium. So again, cryptic refugia would argue against migration. However, if we look more closely at the data, we see that chlorotype, I call it chlorotype gray here, in the paper it's called chlorotype 15. It's the most common chlorotype in Alaska here, and it steadily decreases eastward across Canada. You see this light orange bar here represents the Yukon discontinuity where there's virtually no larch. Whereas the most common haplotype in Canada, the light green haplotype, while there's no geographical trend across Canada, it's fairly common everywhere, but it's virtually absent from Alaska. It's not found in Alaska, this common chlorotype. So these results provide strong evidence for asymmetrical migration across a major barrier, dispersal barrier in Yukon, right? So now let's go to our second tail, the tail of Western red cedar. You see, Western red cedar, is a species from the Pacific Northwest, and its range is divided in interior part of the range and a coastal part of the range, divided by a rain shadow. So our question again is, did red cedar persist in the interior throughout the last glacial maximum, or did it migrate from the coast during the post-glacial? Here we show genetic results from more than 40,000 RADSEC loci from 22 populations. What we see is 4 genetic lineages, a yellow, dark, black, pink, and a blue one, a central blue one. Now, if we look at a PCA, we're looking at each individual, its genetic signature, how they cluster together on multidimensional space. And we see that, well, each individual clusters with each other according to their genetic diversity. And you see that the blue cluster, if you divide each individual, those from the interior in dark blue and those from the coast in light blue, they completely overlap, meaning that they are genetically similar. So despite the fact that there was actually a cryptic refugium in the interior, shown by the black group here, yet most of the interior range originates from asymmetric migration from the coast and not local expansion of the refugial population. Now our last tail, the mountain hemlock, it's a species from the Pacific Northwest. Again, it's divided in an interior range and a coastal range divided by the same rain shadow. And we ask the question, did hemlock persist in the interior or did it migrate from the coast? We see results from 30,000 RADSEC loci from 21 population. You clearly see 2 genetic clusters, orange and teal. And when you look at the PCA, well, what you see is, again, that coast and interior are genetically similar for each lineage, for the teal lineage and the orange lineage. But in both cases, interior, meaning darker colors, are a subset of the coastal diversity. All right, so these are clear indication that the interior range was successfully colonized at least twice through asymmetric migration from the coast. Now, I've presented to you 3 cases where there was successful migration, asymmetric migration from the west to the east. And you can argue, Guillaume, these are wind dispersed trees. Big deal, you've detected the westerlies. Well, I can agree with that. Of course, I wrote it down. But then how to explain these results? Where for, at least for Mountain Hemlock, we found higher wing loading in coastal populations with respect to interior populations. Wing loading is a ratio between seed mass and wing area. And of course, there's an allometry between the 2 threads. But for any given wing area, the seeds are lighter in the interior compared to the coast. So seeds from coastal hemlock population are heavier and have smaller wings. They have less dispersal capacity compared to seeds from the interior. So this introduces the question, is it possible that the contrasted dispersal capacity reflects rapidly evolving intraspecific threats during migration? So here we invoke spatial sorting as a model that we wish to test. It's an emerging eco-evolutionary model whereby our predicting that alleles that confer highest dispersal will accumulate at the expanding front. I summarize this spatial sorting model with these 3 keywords: disperse, breed, repeat. Disperse because individuals start from a fixed point and move away. The ones that move faster will form the new expanding front. Then they breed. Fast dispersing individuals at the expanding front breed with each other simply because slow dispersers are left behind. And this interbreeding at the expansion front will produce offspring with higher mean dispersal rates than the parental generation. And then if you repeat this process over time, successive generations will evolve faster and faster, increasingly faster dispersal. And such cumulative assembly of novel phenotypes are ever more adept for dispersal. And note that this occurs without the need to affect fitness. This evolutionary process is called spatial sorting. It's not natural selection. So let's just take a look at how it can, in a small animation, how it can occur. Here we see an ellipse as an individual, and you see blue dots and orange dots. Blue dots are genes with alleles that confer higher dispersal rate, and orange or brownish circles are genes with alleles that do not confer high dispersal rate. And here you see dispersal. Individuals start from a fixed point, move away. Some individual that moves faster form the expanding front, then they breed together, and those faster individual will breed with each other, and then it will create offspring with higher mean dispersal rates than the parental generation, and then repeating this over time will result in novel phenotypes more and more adept for dispersal. And if you take a look at this animation, you see here that dispersal, according to this model, dispersal is expected to increase over generations, right? So the spatial sorting model implies exponential increase of dispersal capacity during migration. Colonization fronts should be dominated by fast dispersing individual. This is where we should assess the actual migration capacity of species in response to climate change. Not in the core of the range, not in the south, not where we live. We should go up north where colonization capacity is important. This framework needs immediate empirical investigation in trees because they could imply natural solutions to the migration lag in a fragmented world problem. And these natural solutions might be important before implementing costly, risky, and much debated assisted migration approaches for conservation purposes. Assessing the dynamics of range expansion should provide key insight about the real capacity to cope with anthropogenic climate change in those populations where migration response actually matters for conservation, that is at species range margin. So ongoing project in my research team assess the spatial sorting model in altitudinal and latitudinal tree lines of boreal conifer species in Quebec. We work with the expectation that higher dispersal capacity will be found for many species at the colonization fronts at various scales that will reflect post-glacial migration along the latitudinal tree line and current expansion since little ice age and anthropogenic climate change along altitudinal tree lines. And we will investigate dispersal threats, so basically spatial sorting of phenotypes, as well as the genomic imprints, the spatial sorting of genes. And we will test for associations among genotypes, phenotypes, environment, and geography. So with that being said, I thank you very much.